everyone, and welcome to Histology 101, where we talk all things histology, everything you wanted to know about histology. Probably didn't want to, but are forced to. I'm Dr. Jamie Chapman. I'm going to be talking to you today about the histology of ovaries, oogenesis, and folliculogenesis. It's one of those topics that is quite complicated. Reproduction within the ovaries, production of oocytes in terms of meiosis, which we call oogenesis, is quite a complicated thing. And then how does it tie into folliculogenesis? And of course, adding to this complexity is some misinformation that's carried over in a number of textbooks out there for simplicity's sake. So when we talk about the menstrual cycle occurring over 28 days, they often include the ovarian cycle and what's happening within the ovaries over those 28 days. But in fact, what happens within the ovary takes a much longer time. And for simplicity's sake, textbooks tend to cram everything into those 28 days. So I'm going to tell you the truth, exactly what happens. So yeah, let's see how we go. Welcome along, everyone. Good watermelon, nice. Thanks very much for coming along. Just as we start, please let me thank Indri, who followed a week ago, Alcium Blue, who followed 4 days ago, and Argentum Ferritra, who followed 2 days ago. Thank you very much for the follow. It's really great to have you along. Welcome to the slide box, as I like to say. Anyone who sort of follows are a slide in our slide box, and we enjoy having you as part of our group. All right, so it's very difficult to know where to actually start with talking about the histology of the ovaries and oogenesis and folliculogenesis. Maybe we'll start with the histology and sort of break down the normal structure of the ovary, and then we can talk a little bit more about oogenesis and folliculogenesis and what that actually means a little bit separately. And the reason I wasn't quite on time was because I've been trying to find the images that I can use to explain everything, the ones that I like to use in my lectures anyway. All right, so remember, please, any questions along the way, pop them in chat. We've got Dr. James Crane sitting to my side here, who is monitoring the chat, and he can help to answer questions, point out things to me that I might have missed, and so on. So yeah, let's go. Right. So, first of all, of course, let's get, where are we? Here. Okay. All right. So, Blubbery HXG, thanks for helping me in histology revision. You're welcome. Thanks very much for coming along. This is what I love to do. I love to try to make histology easy. I do know it's quite a complicated subject for a number of students that just don't find it naturally coming to them, and I really enjoy teaching it, and so it's one of my joys to actually spend a bit of time talking about it. All right, so really welcome along. It's really great to have you here. This is an odd section, so I might start with another section. All right, we'll start here, okay? We'll start here. We'll move to a more normal section, I say in inverted brackets, and then we'll come back to this one and interpret it, because this one's a little bit complicated. But generally speaking, the ovary is divided into two components. It's an outer cortex and an inner medullary region. So this is the outer cortex we can see here, and then this is the inner medullary region. And the medulla is kind of continuous with the ovarian ligament, which sort of extends out here, contains the ovarian vessels, and then that generally attaches to the lateral sides of the uterine wall. Now, within the cortex is where we find the follicles at various stages of development. So you can actually see some of these large empty spaces here. These are some of the antral follicles, so they're more developed follicles. And then the little holes you can just sort of get a little impression of are some of the smaller follicles stored within the ovaries. Now, on the surface of the ovary, and I'm going to go back one slide here, we'll sort of zoom out. On the, actually, I've jumped ahead. What I wanted to point out is that on the surface of the ovary, we actually have a epithelial lining, and this is a simple cuboidal type of epithelium. This is known as surface ovarian epithelium. So this lovely simple cuboidal epithelium here. So this is known as surface ovarian epithelium, or it's also called sometimes germinal epithelium. Is the germinal epithelium just a continuation of the broad ligament? I'm not sure about the broad ligament, good watermelon, but it's actually a continuation of the peritoneal coverings of the ovary. So normally most peritoneia are a simple squamous type of lining, but for some reason here it's actually a simple cuboidal type of epithelium. So it's considered sort of an extension of the peritoneum on the surface of the ovary. Sort of takes its origin, of course, the gonads begin development near the kidneys in the abdomen, and then during development they migrate their way out into the pelvis for the people assigned female at birth, and then for males, people assigned male at birth. I'm going to have to excuse my terminology here. I'm trying to get familiar with it. The gonads, of course, pass through the inguinal canal and continue on development into the scrotum. So as they do, they drag their peritoneal coverings, they drag their blood supply, which is why the gonads have their blood vessel origin up in the abdomen, which is kind of a strange place if you think about it, because they're quite relatively far removed from that origin of that blood supply. So as I was saying, Chapman family, g'day mate, hope you're well. So I am assuming that is one of my sons watching from home, who's logged into my other account. So who is that? Is that Noah? Hello and welcome along. Thanks very much for coming along. All right, so that was a bit of a distraction. On the surface here, we've got our surface ovarian epithelium, simple cuboidal epithelium. And then we're going to go back to this this other slide, which is a much nicer slide. Beneath this epithelium, we have this thick connective tissue layer here. And this is known as the Tunica albuginea, which just basically means white layer. And interestingly enough, we see that term, Tunica albuginea, come up a number of times in relation to the reproductive tract. So the testis, for example, has a Tunica albuginea, a big thick connective tissue as part of the testis wall. And also in the penis, around the corpus cavernosa and the corpus spongiosum, there's a layer, a dense connective tissue layer there, which is also known as the corpus Tunica albuginea. So that term pops up a number of times. Now, because it's a simple cuboidal epithelium, it's often easily disrupted from the surface of the ovary, so it's not always present in all sections. But the cortex you can see here, this is obviously a section from a relatively young animal, and the reason we can tell that is because of all of these follicles here. These are the first follicles, or the store of follicles that occurs within the ovaries after maturity, and these are known as primordial follicles. Now, primordial follicles are characterized by having a primary oocyte. So this is an oocyte which has already begun to go into meiosis. It's gone into prophase 1 and become arrested. And then it forms this little nucleus. You can see here, nucleolus, here's the nucleus, here's the cytoplasm. So this is a primary oocyte. And around the outside of the primary oocyte of a primordial follicle is a simple squamous lining of follicle cells. Now, they're not always that easy to see. You can just hopefully just make out some nuclei here. These are flattened nuclei. So just one single layer of flattened follicular cells. Now, in between these primordial follicles is the part of the ovary known as the stroma. So the stroma is in between the follicles. So you can see all the stroma here, and of course that's where we'll find the blood vessels, the connective tissue. We often find smooth muscle cells in there as well. But all of these are actually primordial follicles. So they're the store of follicles within the ovaries. Now, at some stage, these follicles get some stimulus, some internal stimulus to actually begin development. So the primordial follicles become primary follicles. And two main things happen when a primordial follicle begins to develop and become a primary follicle. The first thing that happens is the oocyte gets bigger. So here you can see, here's our primordial follicle with the squamous follicular cells. And you can see how much bigger this oocyte is. Its nucleus is here. It's not completely in this plane of section, but you can generally get the idea. Here's the oocyte cytoplasm or the ooplasm. And then the other thing that happens, so we get our growth of our oocyte, we also get growth and transformation of those follicle cells into a simple cuboidal type of epithelium known as the granulosa cell layer. So we start off with one single layer of granulosa cells, and then eventually these continue to develop and form multiple layers. So initially we have one layer of granulosa cells, and then they'll go on to form multiple layers. So this we refer to as a unilaminar primary follicle or a unilaminar pre-antral follicle. So this is an early stage. Its basement membrane would be sitting around on the outside here. And then on the outside, we begin to get differentiation of some of these stromal cells. And then you can see they sort of start to encapsulate the outside, on the outside there. And so some of those stromal cells will differentiate into a new layer, which then subdivides into two further sublayers. And this becomes known as the thecal layer. And the ones closest to the basement membrane are known as theca interna cells, and then external to that would be the theca externa cells. Now the theca externa cells are basically just anchors to the surrounding stroma, and the theca interna cells play a really important role in the hormone production during development of these follicles. And we can talk more about that in a little while. All right. So that's our unilaminar primary follicle. I did see a multilaminar one over here, yep. So here they continue development. The oocyte has continued to get bigger. So I wonder if we can get them in the same plane of section. So here's our primary, unilaminar primary. That then becomes a multilaminar primary, we can see over here. So we zoom in. And so our oocyte has continued to get bigger. We've got two or more layers now of granulosa cells forming the structure of the wall. And let me just say to my children at home, it would be really appreciated if you could stop commenting in the chat. Thank you very much. So here's our primary oocyte. We've got our multiple layers of granulosa cells, and what happens is secreted on the outside of the oocyte is a new layer. This is known as the zona pellucida. Let me see if I can. I'm going to write it with the mouse here. So zona. Oh. I'm going to have to get the pencil out, aren't I? Here, it's really gross. All right, so that's the zona pellucida just here. This little acidophilic band around the outside. Now this is a glycoprotein coat that comes to surround the oocyte, and it's actually a really important component when sperm actually meet the oocyte. All right, so that's the zona pellucida. As I say, this is the granulosa cell layer. We will often describe this as the zona granulosa. So often this has we give this the abbreviation ZG. Zona granulosa. The basement membrane would be here, and then on the outside we have our theca interna and our theca externa cells. Okay, so if that continues to develop, what happens is those granulosa cells continue to divide. They eventually begin to secrete fluid as well, and over time they form these little fluid-filled spaces. So a primary follicle must have multiple layers of granulosa. No, Ruby. So you can actually have two types of primary follicles. You can have a unilaminar primary follicle, or you can have one of the multilaminar. So it can either be one or two layers or more layers. So it can either have a simple epithelium or a stratified epithelium. Which cells are responsible for the production of the zona pellucida? Well, good watermelon, that's a bit of a controversial question. I actually did my PhD on the zona pellucida of marsupials, and it seems to be a little bit of a debate in the literature. I think it's almost exclusively from the oocyte secreting the zona pellucida around the outside. There does seem to be a little bit of evidence out there that it may be get some contribution from the granulosa cells. So it's either the oocyte, the granulosa cells, or a mix of both. My personal feeling is likely going to be the oocyte that secretes it. We can have a big discussion about the literature another time. So our multilaminar then continues development. I'm trying to find a small antral follicle if it's possible, so we can show a sort of an early secondary. I don't think we have one in this section. There might be one in the next this other section here. This. All right, so we can sort of get a little bit of an impression here. It's a little bit disrupted. It doesn't look particularly nice, these follicles over here, but I'm just zooming around. Yeah, so the follicles then begin to secrete fluid, and eventually that fluid, which is known as follicular fluid, eventually fills this one space. So all those little pockets of fluid that initially form begin to fill into one space, and this is known as the follicular antrum. Okay, so at this point we recognize this follicle as an antral follicle because it's got a fluid-filled space. So here's the oocyte. It's looking very odd here. I think there's a little bit of disruption going on. And then we've got our fluid-filled antrum. We then have our zona granulosa, our basement membrane, and then our theca interna and theca externa on the outside. Now, so depending on the textbook, the terminology changes in terms of classification of follicles. So I classify this one as a secondary follicle. Some people actually classify this as a tertiary follicle. So they classify a primary as a unilaminar primary, a secondary as a multilaminar primary, and then a tertiary follicle as an antral follicle, and then of course a pre-ovulatory follicle as the big, large Graafian follicle. But you know, whatever terminology you use, this is an antral follicle, and this is about the level a follicle will develop without the presence of follicle-stimulating hormone. So, in order for a small antral follicle to develop any further to become a larger pre-ovulatory follicle or a larger antral follicle, it requires follicle-stimulating hormone. So all of this development that we've been talking about so far, the development of a primordial to a primary, to a multilaminar primary, and then to an antral follicle, this actually doesn't require, this early development doesn't require FSH. It's a process which we call gonadotropin independent growth. So pre-antral follicular growth is considered follicle stimulating hormone independent, doesn't require FSH. What does require FSH, and what we consider something called cyclic recruitment, where we get capturing of already developing antral follicles, is FSH, and that of course only occurs after puberty. If we were to take a section of the ovary of a 8-year-old girl, we would see lots and lots of primordial follicles like this. We'd see primary follicles and we'd see small secondary follicles, but we wouldn't see anything beyond that because FSH levels, circulating levels of FSH aren't in very high levels, and therefore there's nothing to capture those follicles to continue to them grow. So those follicles continue to get bigger and bigger. You can see the big fluid-filled space here. We've got this nice thick wall here of the zona granulosa. Here, the basement membrane would be here, and then the theca interna cells would be on the outside here. Now, the theca interna cells are important. They have receptors for luteinizing hormone, and in the presence of luteinizing hormone, they actually produce an androgen. And then that androgen is picked up by these granulosa cells And these granulosa cells have FSH receptors. And so FSH causes them to actually change the structure of that androgen in a process called aromatization. So they convert this androgen into an estrogen through the process of aromatization using the enzyme aromatase. And so as these follicles are captured, and as FSH is capturing them, what it's doing is it's starting to cause them to continue to grow, and the levels of estrogen begin to increase. So it reaches a point where those estrogen levels are high enough, and actually moderate levels of estrogen actually has a sort of an inhibitory effect on FSH and LH secretion. So it's really a strange situation. It actually has both a stimulatory and an inhibitory effect. So initially, this levels of estrogen has an inhibitory effect on FSH release, but then once we get our pre-ovulatory follicle selected and that becomes the dominant follicle, then that's such a large follicle that it's producing lots and lots of estrogen. It reaches a peak, and then that actually has a positive feedback effect on the release of GnRH, which then subsequently has a release on LH particularly, that eventually leads to ovulation and the formation of the corpus luteum. So over here, we've actually got this nice secondary follicle here. So here we've got our oocyte. Here's where our zona pellucida should be. Hasn't quite stuck around very well here. And then you might notice that we've got this layer of cells on the outside of the oocyte and the zona pellucida here. Now, they're sort of modified granulosa cells, and these become known as the corona radiata cells. And the cells which sort of attach these cells here, which attach it to the wall of the follicle, the zona granulosa, are known as the cumulus ooiferus. I better write these down because they are a little bit complicated. So let's get our little label here. So here's our primary oocyte, still arrested at prophase 1. This is our zona pellucida. This is our corona radiata. So the corona is like the sun, the corona of the sun, and radiating out from the zona pellucida here. So this is the antrum. This is our zona granulosa. And here, this region here, this is known as the cumulus ooiferus. So the cumulus ooiferus sort of attaches the corona radiata to the zona granulosa, if you like. There'd be a basement membrane here, and then we've got our theca interna cells and theca externa external to that. So as I mentioned, these cells have receptors for luteinizing hormone. These have receptors for follicle stimulating hormone. This produces an androgen. And then these produce an estrogen through the process of aromatization of that androgen produced by the theca interna cells. Is the pink center the nucleus or the cytoplasm? So this is all the cytoplasm. So in this plane of section, the nucleus hasn't been sectioned or isn't present in this section. So if there was a nucleus, it would sort of be fitting around this sort of area here. So this whole thing is cytoplasm. Okay. All right, so that continues to develop. In the presence of FSH, it then becomes this large Graafian follicle, and eventually it sort of becomes quite eccentrically located in one side of the large Graafian follicles. You can see how it's almost eccentrically located here. Now this sort of continues to grow. It bulges from the surface of the ovary. We get that large amount of estrogen, which brings about the positive feedback effect on LH secretion, and that causes a thinning of the follicle wall. And this fluid, remembering it's still held by this basement membrane follicle, and so there's all this hydrostatic pressure building up in this follicle, all of that follicular fluid. And then when you get a thinning of that wall, you just need a small little bit of thinning, and then it pops like a balloon and out comes the oocyte onto the surface of the ovary, ready to be picked up by those little finger-like projections of the fimbriae at the end of the uterine tube. And we can actually see those in this, section here. So if we go here, so here's our ovary, and then look at all these strange structures sort of reaching out here, a little bit like coral or something. These are the fimbriae of the uterine tube. So here's the actual uterine tube on the right hand side here, and this is the infundibulum, the end of the uterine tube with its finger-like projections, the fimbriae. If we sort of zoom in here, we can actually see some cilia on the surface. So these are highly ciliated area. So they create a current to draw the oocyte from the pelvic cavity inside the uterine tube for transport down the uterine tube, hopefully for fertilization if that's the what the person who's ovulated wants. All right, so then what happens is that the remnant follicle, we'll go back to this other one here. The remnant follicle then transforms into a corpus luteum. So let's zoom in here. We'll have a look at this one. So these are the zona granulosa. These are the theca interna. So what happens is after ovulation, blood usually comes into this antral region. It forms a large body called a blood body or a corpus hemorrhagicum, and then the basement membrane breaks down, and the granulosa and the thecal cells begin to intermingle with each other, and as they do, they transform in structure. Particularly the zona granulosa transform from a more protein-producing appearance to a more steroid-producing appearance. So they get lots more smooth endoplasmic reticulum. They gather cholesterol from the bloodstream, and so they transform into cells known as the granulosa lutein cells. The theca interna cells transform into the theca lutein cells, and they both begin to secrete progesterone, which begins to increase in levels as this ovulated follicle transforms into a corpus luteum. Now, so the theca interna still play, the thecal cells still play that role of producing an androgen, which then is converted into estrogen, but they also both then actually produce progesterone. So we see an increase in both progesterone and estrogen after the formation of the corpus luteum. Now, I want to show you this slide. As I mentioned, it's a little bit difficult to interpret. So here's the main part of the ovary here, and then we've got this weird sort of floating bubble sitting on top here, which is almost as large as the ovary we've got here. Now it looks like it's actually not connected, but this is actually ovarian tissue as well, and this very large structure here, this is actually a very large corpus luteum. If we zoom in here, look at these cells, aren't they beautiful? The big pink puffy cells, like fairy floss or something like that. So here's the nuclei. We can see all of these blood vessels. So these are granulosa lutein cells and theca lutein cells of the corpus luteum. And so these are pumping out lots and lots of progesterone and estrogen. Now initially that's about preparing the endometrium for implantation. But after pregnancy, that corpus luteum is maintained by a hormone secreted by the embryo called human chorionic gonadotrophin, and that comes from the trophoblast cells, and that maintains the corpus luteum for the first 3 months of pregnancy. And so what this is actually what I think is a corpus luteum of pregnancy, this massive endocrine organ pumping out large amounts of estrogen and progesterone to maintain that endometrium while we're actually getting implantation, fetal development, the formation of the placenta, and so on. After those 3 months, the placenta itself begins to secrete progesterone and estrogen, maintains itself, and then the corpus luteum can degenerate. Now, when the corpus luteum degenerates, it forms a structure called a corpus albicans, which is sort of like a little scar tissue. And we'll just have a quick look to see if we can see one in this section. It's a very slow load. Can't see one there. So we're going to skip forward. All right, so here's a nice corpus. So here's a corpus luteum. This is sort of the typical sort of appearance of a corpus luteum. So you can see where the antrum was. You can see the blood cells in here. So you can see the corpus hemorrhagicum begins to break down. And then we've got these very acidophilic cells here of the corpus luteum. We often get this very sort of folded nature to the corpus luteum. And so some of these darker cells would be the thicker lutein cells, and the larger, more acidophilic pink cells would be the granulosa luteum. Now, if there's no fertilization, there's no HCG secreted, then that corpus luteum degenerates. And it eventually forms this little bit of scar tissue here, which is known as the corpus albicans, which means sort of white body, because eventually these cells break down, they form scar tissue, collagen, and sort of connective tissue fibers, and therefore it appears white in appearance. There's sort of another one here. You can see the very pale regions. So these are the corpus albicans within the ovaries. And so that probably what happens eventually to the corpus luteum of pregnancy as well, eventually becomes a corpus albicans. All right, so really that's kind of the histological structure of the follicles. So we can have a look at some other examples here. So we sort of zoom in. So we can sort of make out a little bit of that surface ovarian epithelium. Here's our Tunica albuginea. We've got all of these primordial follicles, maybe an early primary one here. You can see how the cells are a bit more rounded here, whereas these are a bit more squamous. If not pregnant, the corpus luteum maintains endometrium for a period of time. Yeah, so sort of the corpus luteum drives, if we talk about the menstrual cycle, occurring over 28 days. If we just sort of... All right, so if day 14 is here, and we talk about the menstrual cycle occurring over these days, the first 5 days is of course menses. Then we have the proliferative phase, and then we have the secretory phase. Okay, so this is the menstrual or the uterine cycle. So this part of the menstrual cycle is driven by the corpus luteum. So the secretory phase is driven by the corpus luteum around about 14 days to, so this is going to be day 28. So around about day 25, 26, something like that, without the presence of the trophoblast layer, because it takes about 6 to 8 days for the blastocyst to form and reach the uterus for implantation. So it's about that stage when we get differentiation of the embryonic cells to form the trophoblast and form the HCG. If no pregnancy has occurred at this point, then the corpus luteum degenerates and we lose our progesterone and estrogen, and then that actually stimulates or causes constriction of blood vessels within the endometrium and leads to the menstrual phase. So even if not pregnant, hopefully Ruby, that's answered your question. Good Watermelon's asked, does that mean after menopause almost the entire ovary is replaced by scar tissue or many corpus albicans? There are a lot of corpus albicans in a ovary of a person, an older person who's actually gone through menopause, because of all those ovulations and the formations of the corpus luteum. And certainly menopause is brought about by the loss of primordial follicles from the ovary. Oh, thanks very much, Booze Days Gone. I'm glad my videos help you out. All right. So, that was the menstrual cycle. Hopefully, Ruby, that answered your question about the corpus luteum and when it sort of breaks down. Booze Days Gone, thanks very much for the follow. I really appreciate that. what we'll go back to have a look at the histology of the ovary again, just to sort of point out and just revise a few of those things that we've talked about to help your interpretation. Okay. So one of the other things I wanted to talk about is this idea of follicular development, so folliculogenesis. Now, there's this lovely diagram which was published a number of years ago, which talked about this concept of initial recruitment. Great, thanks very much, Ruby. Initial recruitment versus cyclic recruitment. So this is where I was talking about the idea of gonadotropin independent growth versus FSH being required for further development. So I don't know if I'm going to be able to draw on this. I might only be able to point things out. Oh, I am. Good. Okay, so initial recruitment versus cyclic recruitment. So basically, cyclic recruitment is what happens after puberty. So from puberty onwards. This occurs from before birth all the way to menopause. So we've sort of got a dividing line here of what happens within the ovaries. So we know that our store of follicles within the ovary are these primordial follicles, and they get some sort of stimulus, internal ovarian stimulus, to begin development. And this is what we call this initial recruitment. They grow, they become primary follicles, they become secondary. So here the terminology is a little bit different. So here this author refers to secondary as being multilaminar primary. Then they become antral follicles, so small antral follicles, or what we were calling early secondary follicles. But without FSH, which there really isn't very high levels of before puberty, so there's no FSH, there's nothing to cause the further development of those follicles, and they all undergo degeneration, which is a process called atresia. Now, atresia is such an important concept to have an understanding of. So we can actually see that these numbers of primordial follicles have actually declined over that time leading up to puberty. Okay, so we actually reach puberty. Puberty, now we have FSH entering circulation. So that's actually capturing some of these already developing antral follicles. It causes one to become a dominant follicle. The others that were captured and begin development but haven't been selected as the dominant follicle undergo atresia. So these follicles are undergoing atresia. Eventually that one dominant follicle gets stimulated to undergo ovulation, and that happens once every month normally, over a lifetime of a person with ovaries. And around in a person with ovaries lifetime, they may ovulate 400 to 500 oocytes. They actually start with 2 million. They start at birth with about 2 million follicles within their ovaries, and then they actually only ovulate about 400 to 500 over their lifetime. And then eventually, around about 50 years of age, we get depletion of our primordial follicles, exhaustion of the follicles. Oh, you'd be exhausted too after surviving 50 years. And then what it means is that actually we've lost all the follicles within the ovaries, and then that the person enters into menopause. Okay, so FSH is required for cyclic recruitment. Initial recruitment doesn't require FSH. It just is this internal ovarian stimulation that causes, basically causes the primordial follicles to become primary and antral follicles. They actually undergo atresia. Now, this is a really interesting question, because often after a lecture, I give a lecture about this, someone will come up to me and say, what about a person who goes on the contraceptive pill? They don't ovulate. Does that mean that they undergo menopause later because they're actually saving those follicles from being ovulated? Because that's what the contraceptive pill generally works. It prevents ovulation occurring. Generally, they're progesterone based. They may be a mix. Sometimes you can have moderate levels of estrogen based, depending on how the person taking the contraceptive pill deals with those hormones. But generally speaking, they work in preventing this cyclic recruitment from taking place. But now that we actually know what's going on within the ovaries, we know that initial recruitment's happening whether the person's on a contraceptive pill or not, because this isn't influenced at all by FSH or LH or GnRH, gonadotropin releasing hormone, which is driving FSH and LH secretion. So a person on the contraceptive pill, while they are no longer undergoing cyclic recruitment, they can't stop initial recruitment. That's still taking place within the ovaries. So they're still getting primordial follicles becoming primary, becoming small secondary follicles, but without that FSH secretion, those follicles are still undergoing atresia, and this person taking the contraceptive pill, generally speaking, doesn't have any delay in menopause, unfortunately. Now, if someone could develop something to stop this initial recruitment, then that might be something worth looking at. You could actually delay entering into menopause. It might be something worthwhile. I know a lot of people out there would like to delay having kids, and the idea of this concept of the ticking clock, biological clock. And there is a fertile period where after a while, after a certain age, then ovulation becomes less regular and becomes much more problematic for people to actually get pregnant and so on. That's a little bit different of a histology talk, isn't it? That's this idea of folliculogenesis. Now, what I wanted to point out from discussing this was that atresia is an important process that takes place within the ovaries. So when we actually look at the structure of the ovaries, atretic follicles are a major component of that. Now, how do we tell an atretic follicle from a healthy follicle? Well, some of them are going to be pretty easy to spot because they just look really unhealthy. So what you look for is look at the zona granulosa. Are all of the cells really nicely, tightly compacted together, and so on, or are they quite disrupted? Sorry, my mouse cord was stuck there. So you can see these are actually beginning to separate from each other. So I think this follicle here is probably undergoing atresia. But we can actually see evidence of some of these previous atretic follicles. So we often refer to these little glycoprotein things here as zona ghosts. They're kind of ghosts of oocyte past, if you like. These were follicles with oocytes within them, and they've undergone degeneration, and you get what remains behind is these little scar tissues. And these are old atretic follicles. You can sort of see some of these over here. So atretic follicles and identifying them are actually part of the histology of the ovary. We don't really talk about it very much, funny enough. You don't really hear about it very much in textbooks, but you can see it's quite a major feature of the histology of the ovaries. Atretic follicles still have an antrum. Well, of course, depends on what stage of atresia they're at. So some of them may have. Actually, let's go back to one of these other sections. This one, there's a really nice picture here. Yeah, side by side. On this left here, we've got an atretic follicle or a follicle undergoing atresia, and we've got a healthy follicle on this side here. So look how nice and healthy these zona granulosa cells look like. Nice, tightly compacted together, whereas these cells, look how disrupted they are. There's lots of space in between them. You can see that they've started to come away. We've still just got some basal cells here, but most of them have started to break off. So side by side, we've actually got a nice, healthy looking antral follicle and a antral follicle which is undergoing atresia. And same over here as well. Look at the zona granulosa here. Very disrupted. We've got quite irregular looking chromatin in the nucleus here. So these cells are breaking down. This is an atretic follicle. Okay. Yeah, so sometimes you can see different appearances. Here's definite atretic follicle. See how it's all breaking down and it just looks really odd. Not that nice, healthy looking zona granulosa there. So atretic follicles can be at any stage of development. They can be antral stage. They can be pre-antral stage, but some of the ones I was showing here were antral follicles undergoing atresia, good watermelon. So hopefully that helped you out. And by the way, thank you very much for the follow, Good Watermelon. I'm really happy to have you in our slide box. You've slid into our slide box as a slide. All right. That's a atretic follicle there. All right, so I think that's pretty much it in terms of the structure of the ovaries. What I did want to talk to you about a little bit is the development of follicles and the idea of oogenesis, or what happens to oocyte numbers. So this, I use this graph in my lecture when I talk about what happens within the ovary, and we've already alluded to some of this here. So hopefully you can see, I'm sorry, I had to use this, I had trouble getting this picture to work on my browser where I could actually talk about it. So I apologize that we've got these weird borders and everything, but hopefully you can see down the bottom here we've got age, so starting at fertilization up until birth, and then from birth onwards to 50 years of age when generally a menopause occurs. And on the left-hand side here on the y-axis, we've got numbers of germ cells in millions. So we're talking about the germ cells. There could be a number of different germ cells which exist within this. So, that's not going to work. All right. That's not going to work. All right. Yep, that's not going to work. All right, so we're not going to draw. I'm just going to have to point things out. All right, so within the developing fetus, we get the development initially, very early on actually, of a type of cell known as primordial germ cells. I might just do the other one then. Let's do this. So we'll clear this. So we have we start off with these primordial germ cells. And sometimes these might be referred to as gonocytes. So this is sort of before differentiation, before we get the presence of a Y chromosome or not, stimulating differentiation of the germ cells and the reproductive tracts. So eventually they then form oogonia, and that developing gonadal ridge becomes a gonad, and then either is directed to become an oogonium or a spermatogonium. You can download the image and draw it on the Windows Photo app. Yeah. I did try that. Let me have a look. So, can we zoom in? And what about annotation? Edit, create, edit, draw. Okay. Ooh, nice. How do I delete that? Is that a rubber?
Yeah, that one.
Yep. All right. So, can we zoom in? Doesn't seem to allow me to zoom in. All right. Anyway, so we get this sort of differentiation of these primordial germ cells. Ooh, okay. All right, I'm learning as we go. Oh, too far. I apologize. All right, let's get rid of that. Why did you run me out? Erase all ink. All right. If inconvenient, perhaps you could do it in Paint as well. Yeah, I haven't really had a lot of play with Windows. I'm more of a Mac person, so I apologize for that. So, if anyone knows how we can zoom in, here we can. View actual size. No. Nah. Anyway, all right, that's another time for me to play around and work out how to zoom in. So we get the primordial germ cells differentiating into oogonia, and then those oogonia undergo this massive amount of mitosis. So we can see them before birth rise in number up to 7 million. There's a magnifying glass above the... Well done. Awesome. Thank you very much. Stimulate my follicle. How appropriate. All right. So we get this massive mitosis of these oogonia in this region here. So oh, it's not letting me draw on here now. Why aren't you letting me draw? Hold control. Okay. All right. Oh, that's so bizarre. And create, draw. All right. Okay, so it's really not allowing me to do both at the same time. So these are oogonia. Oh, come on. All right, this is really good strain. All right, so oogonia undergo lots and lots of mitosis, all right? And then we actually get some die-off of those oogonia. So we actually have several processes going on. One of those is apoptosis. And this is sort of a throwback, evolutionary throwback to when we used to be external fertilizers. So it made sense. We made lots and lots of oocytes. They were all released at once into the sea, and then they were fertilized externally. And then when we evolved to move everything internally, everything sort of moved inside, and so we actually get this massive die-off, this apoptosis happening here. Now, at some stage during this development, so we're still fetal here, with this still within the developing ovaries of the fetus, all of those oogonia actually begin meiosis 1. So they become primary oocytes and they enter into meiosis 1. And they become known as primary oocytes. And they arrest at prophase 1. Sorry, let me get rid of this now. So all of the oogonia, so this is still before birth. Oogonia all enter into meiosis 1. They arrest prophase 1, and they become known as primary oocytes. Okay, so primary oocytes arrested at prophase 1, and it's this point that birth takes place. All oocytes within the ovary at birth are within primordial follicles or beyond, and they are all primary oocytes. So primary oocytes, they may be in primordial follicles, or they may be in primary follicles. Now, around birth, because it takes around about a year for a follicle to develop from a primordial to a primary, at birth, of course, we wouldn't find anything beyond primary follicles, but we can find, say, in a 2-year-old girl, we might find early secondary follicle or two. Okay, so this is what we find within the ovary. So these, again, primary oocytes within primordial follicles, within primary follicles, within secondary follicles. So this, of course, is because of that idea of initial recruitment. So it's not until puberty that once a month we get this surge of luteinizing hormone, which brings about resumption of meiosis. So a primary oocyte then becomes a secondary oocyte. Now we know in normal sort of cell division we go from one cell to two cells, but here rather than going from one cell to two cells, we go from one cell to one cell, which is a secondary oocyte, plus a little membrane-bound bag of chromosomes, and this is known as a first polar body. Now this is only going to be found within a Graafian follicle or a pre-ovulatory follicle. Oops, sorry. Graafian or a pre-ovulatory follicle. So this is the point at which ovulation takes place. And this secondary oocyte is arrested again, this time at metaphase 2. Okay, and that's the point at which ovulation takes place, and that's as far as it'll go, unless we have a sperm coming to fertilize that oocyte. So meiosis, well, this is the whole process which we call oogenesis. This is meiosis within the ovary. We moves from an oogonium becoming a primary oocyte, that becomes arrested at prophase 1. It's housed within primordial follicles, primary follicles, secondary follicles, and then once a month, following a surge of luteinizing hormone, that primary oocyte is stimulated to resume meiosis 1, prophase 1, metaphase 1, anaphase 1, telophase 1, and then enters and forms a secondary oocyte, which immediately enters into meiosis 2, prophase 2, metaphase 2, and that's where it arrests again. Now, following that division, secondary oocyte, it spits out a first polar body, and then that actually goes on to degenerate. So if you ever see in the textbooks that the first polar body divides, that's just rubbish. It doesn't actually have the cellular machinery to actually undergo cell division. It's a little bag of chromosomes. It doesn't have centrioles. It doesn't go through all those complex processes in order for cell division to take place. What people have misidentified as cell division is actually degradation of those first polar bodies. All right, so this is what's ovulated, a secondary oocyte arrested at metaphase 2, a first polar body, all housed within a zona pellucida and a corona radiata. And that's the point at which ovulation takes place. And that's where the little fimbriae pick up the oocyte and so on. This is as far as it'll go unless fertilization takes place. Now, if fertilization takes place, then we actually get this surge of calcium inside the cell, and then that brings about resumption of meiosis, metaphase 2, anaphase 2, telophase 2, and we get our fertilized oocyte.
You look like you've got a sperm swimming through your head, Jamie.
Don't eat the sperm.
Yeah.
All right, let me go screen only. All right. Secondary oocyte found in Graafian follicle only. That's what's ovulated. Yes, exactly, Ruby. That's what's ovulated. So a secondary oocyte arrested at metaphase 2. It's got a little first polar body in its perivitelline space, surrounded by a zona pellucida, and then surrounded by several layers of corona radiata cells. So those are the barriers which sperm need to get through in order to fertilize the oocyte. Corona radiata, zona pellucida, and then it reaches the oocyte cell membrane. So it's binding of the sperm membrane to the oocyte cell membrane that induces that calcium influx and causes resumption of meiosis 2, metaphase 2, anaphase 2, telophase 2, and we end up with this fertilized oocyte. That's okay, it's sort of gone all, there's something about Mary, I get sperm in my hair. So when the first polar body is degenerated, how many pairs of chromosomes remain? Okay. Primary oocytes are 2N, but of that duplex chromatid type of chromosomes. Okay, so we've got the homologous chromosomes. What meiosis 1 does is separates those homologous chromosomes. So our secondary oocyte is N, haploid, but still with a single of those homologous, the duplex chromatid type of chromosomes. And then this resumes separates the chromatids, the fertilized oocyte will have 23 single chromatid type of chromosomes. What the fertilized oocyte does as well, it spits out a second polar body. This is getting really messy, isn't it? A second polar body. This is also N with a single chromatid type of chromosomes. So our secondary oocyte has 23 chromosomes, but of the duplex chromatid type, so there's still 2 chromosomes stuck together. Should fertilization take place, it resumes meiosis 2. Meiosis 2 separates those chromatids. We get 23 single chromatids going to the fertilized oocyte, 23 chromatids going to our second polar body here. And again, that also degenerates like the first polar body over time. And so, of course, our sperm now is contributing 23 single chromatid chromosomes. 23 plus 23 is 46. We've got our zygote, okay? So luteinase me father 483. So when the first polar body is generated, how many pairs of chromosomes remain? So I'm not exactly sure what you mean in terms of remain. Do you mean remain inside the secondary oocyte, or do you mean within the first polar body? I'm assuming you mean within the secondary oocyte. So here, as I say, 23 haploid chromosomes, 23 chromosomes, but of the duplex chromatid type. They don't undergo that separation of those chromatids unless fertilization takes place. Is that what you meant, luteinase me father? That explanation clarifies it. Great. Well done. Excellent. All right. So I know that's really messy. Hopefully it sort of helps explain what happens during oogenesis. Are there any questions about that? As I say, it's quite complicated. You can see that folliculogenesis is kind of relevant, but it's really not also relevant. So it's to the side here. Oogenesis is mainly meiosis, so it's talking about the oocytes. Folliculogenesis is talking about the development of the follicles and sort of separate from what happens with meiosis. Yeah, I think that has pretty much covered the histology of the ovaries, the structure and function of folliculogenesis, and the structure and function of oogenesis. Are there any late questions? Otherwise, I notice that's really it. Is the first polar body also housed within the Graafian follicle where it degenerates? Okay, so this might be, I'll just quickly save this, and might be worth, all right. So, I didn't mean to close that. All right, so here's our... actually, that's probably not a good...
So while you're drawing that, Jamie,
Yeah.
there's a question from Good Watermelon as well. You might sort of be able to answer it as you draw.
Yep.
So,
About
so the question is, are
tight
there tight
junctions.
junctions between the zona granulosa cells forming something like the blood-testes barrier, so some sort of equivalent?
So the literature again is a bit divided. Theoretically, I don't expect that there would be. Because if we think about it, why we have a blood-testis barrier is that the secondary spermatocytes are genetically different from the male within which they're being produced because of that crossing over event that happens during prophase 1. But if we think about the structure of the ovary, we know that it's just made up of primary oocytes, except for one a month that becomes a secondary oocyte. So that one completes meiosis 1 and therefore is genetically different. However, so that means then all of the oocytes within the ovary, except for that one a month, is a primary oocyte arrested at prophase 1. It hasn't finished crossing over, it hasn't undergone division, so it's still a self-cell. So genetically speaking, it shouldn't need to be hidden from the immune system in the same way as the the secondary spermatocytes onwards. Also, if we think about this what we're drawing now, this is the ovulated oocyte. So here's our secondary oocyte, arrested at metaphase 2. So these are the chromosomes aligned along the metaphase plate. So there might be our meiotic spindles there. So metaphase 2. This is known as the perivitelline space. And here is our first polar body. This is our zona pellucida. And then this is our corona radiata. Okay, so this is what's ovulated. So Ruby asked me about, is the first polar body housed within the Graafian follicle where it degenerates? So it's all encased within this little self-contained unit of this ovulated structure. So ovulated structure includes the secondary oocyte, its zona pellucida, its corona radiata. So the first polar body is carried with the ovulated oocyte, and then should fertilization takes place, eventually you would get a second polar body extruded as well. You'd have the sperm nucleus being incorporated and so on. So they're also carried while the cell undergoes cell division during early embryonic development, from the zygote to the two blastomeres and so on and so on. So they're carried around, and you can actually often see those polar bodies right up until the embryo, the blastocyst, is ready to implant 6 days later. So this is quite a slow degeneration process that takes place. Now, getting to the tight junctions of the zona granulosa. As I say, all of the primary oocytes are actually not genetically different yet. They haven't undergone meiosis 1, so they're not genetically different. When they are genetically different, like the secondary oocyte, which part of the cells are actually recognized as foreign? And of course, it's the cell membrane. This is the cell membrane of the oocyte. If you're an immune cell out here and you're trying to recognize this oocyte as being foreign, look, this cell actually is encased within several layers of cells in the forms of the corona radiata cells. Then you've got this thick glycoprotein barrier. So in order for it to be recognized as foreign, it's got to get rid of all of those extra layers, and it's actually hidden from the immune system by having these multiple layers on the outside. So theoretically, I don't think there would be, and I know in the literature there is this idea of a follicle ovary barrier. I don't really understand why it would need it. So it doesn't have, as far as I'm aware, it doesn't have the same robust tight junction follicle barrier like the blood testis barrier. Good watermelon. Thanks. Glad my lecture finished early so I could watch the live. Perfect for my exam next week. SkinFit, welcome. Thanks very much for coming along. We're just wrapping up, unfortunately, unless you have sort of any last minute questions. Thank you, by the way, SkinFit, for the follow. I just noticed your follow there as well. Follicles are the structure that houses the oocytes. That's correct. So that terminology becomes really tricky. I know a lot of students, when I'm marking exam questions, they talk about the secondary follicle being ovulated and picked up by the fimbria of the uterine tube, but of course it's the secondary oocyte which is ovulated. What ruptures is the Graafian follicle and the pre-ovulatory follicle that then becomes the corpus luteum. So terminology between oocyte and follicles does get a little bit tricky, which is why it's helpful to go back to this and have an understanding of that primary oocytes are only found in primordial, primary, secondary, even early Graafian follicles before that LH surge, and that secondary oocytes are only ever found in Graafian follicles and then outside in the uterine tube following ovulation. Yep, thanks James and Jamie. All right, well, that pretty much does it for us here for this stream. I hope you found it useful. I love talking about this stuff. As I say, this is sort of my background. My PhD work was in reproductive biology. In our next stream, we'll have a look at the reproductive tract of people assigned female at birth, and we can talk about what actually happens with the ovarian cycle and the follicular cycle, the ovarian cycle and the menstrual cycle, how the hormones from the ovarian cycle drive the menstrual cycle, and we can have a look at how the hormones change the various organs within the reproductive tract of people assigned female at birth.
They can't see you, Jamie. You're talking.
I'm talking and I'm not here. Let's go to the camera. Hello. Yes, luteinase, absolutely. We'll do a stream on the testis as well. We'll definitely talk about that. So we can actually allow that question.
Yeah, me too. Yeah.
So apparently testis, you're not allowed to say testis in our chat, which makes talking about reproductive biology really tricky. Wait until we start talking about penis. But apparently it's okay. I can say sperm. Maybe I shouldn't. Maybe there's a term of service in Twitch where I'm about to get banned. You're very welcome. I'm glad you came along. It was really great to have actually interaction in the chat. So I really appreciate it. Hopefully most of that made sense. If you have any questions, hit me up on Twitter. I think you can find the link to my Twitter in the description for the Twitch channel. Otherwise, it's at Chapman _histo. So yeah. We'll look for another time next week where we can continue our streams. Thank you, everyone. Yeah, tell your friends. The more the merrier that come along. It's great to have everyone here. All right, so we'll see you in the next live stream. Thank you, Blubbery HGXG. It's great to have you along. We'll see you in the next stream. Have a great weekend. Hooroo! We'll shout out to my Histo class at U Sydney. Thanks, SkinFit. That's really great. All right.